Hi everyone, welcome to week 9. So we have basically just 2 weeks left of new material introduced in this course before we take our cumulative final exam. So these last 2 weeks are busy, but not as content rich. We're basically finishing off some of our projects. So let's take a look at what we will be doing this week. So we will be starting our final topic of the term, which is evolution by natural and artificial selection. So I'm going to go through some of the information here. So first off, you do have a final exam proctor session sign up that you'll be completing this week. I have it hidden right now because I haven't fully decided on the sessions that I want to proctor. But once this becomes available, do take a look at your schedule for final exam week and choose a time that works best for you. I try to keep them a little bit later in the week because I know a lot of students prefer to have as much study time as possible. And then I try to keep a variety of times, so in the morning, in the afternoon, in the evening hours. Okay, so choose the session that works best for you. If you know that you cannot make any of those sessions, then you might want to go to ProctorU, that website, and sign up sooner rather than later for a proctored exam through them. That way it costs less. If you wait till last minute, it actually costs more to get ProctorU to find somebody to cover your proctoring session. Okay. We'll take a look at our weekly due dates calendar for this week. So we're going to begin topic 8 assignments. There's actually 3 of them this week, and that's it for the entire topic. But we'll talk about why we have it continued into next week as well. So you'll begin working on them. We will be finishing topic 7 this week, so with a quiz. So hopefully you will see the feedback that I've provided on our part 2 assignments by tonight, and then you can relearn any information that maybe you misunderstood, or review what the answers for other students' postings were and so forth. And that will help prepare you for the topic 7 quiz. Now remember, the topic 7 quiz covers all of topic 7, so it's 2 weeks worth of material. Okay. And then we have our 3 assignments this week. Now notice the timing is a little different than normal, and that's because we have some major projects coming up that we want to make sure that you have plenty of time to work on. So your first assignment, topic 8, step 1, is going to be due at our normal time, Sunday midnight. And then the other 2 assignments, step 2 and step 3, are going to be due by Tuesday next week. Okay, so it's a little different. And the reason for that is because we have an integration project due on Monday. And we want to make sure everybody posts this on time because everybody is going to be reviewing each other's posts. Okay, and then don't forget, we still have one final lab report, and that's going to be due not this Friday but next Friday. And then we do have a extra credit assignment that is available starting next week. We'll talk about. Okay. So as always, good idea to review what you learned in topic 7 with Dr. Blackwell's video, and then he'll kind of introduce you into the topics covered in our final topic, topic 8. We have our reading assignment, so that will be from our Edigia textbook. Let me go back and open it up. Okay. And so in our Edigia textbook, we're going to be looking at topic 8. So we do have I believe some questions from this topic that we'll be going through. There's 3 questions, it looks like, in this topic. So do make sure that you've answered all of the questions before you take the quiz. And that'll give you your final grade for your Edigia readings. So remember, in your grade book, there it is 55 points for the answering the questions within the textbook. So I try to update those weekly, but you'll get your final grade soon on it. Okay. Going back into our topic 8. So let's take a look at the specifics of topic 8. So we have our final student roles. So, figure out which student number you are. And then let's check out the first step of our topic 8. So in the first step, we're going to be taking a look at genetic diversity, fitness, and natural selection. So you may need to review your textbook reading before you actually start working on this to just get an idea of what terms adaptation, fitness, that kind of thing, what they are, and then just understanding what natural selection is are important for you in answering the questions. There's also a mini lecture, okay, so this just kind of goes over some of the information that's been kind of summarized for you from the chapter readings. Okay. So once you feel confident about your understanding of genetic diversity, fitness, and natural selection, then you'll go ahead and apply that to the questions, starting on page 299 all the way through page 304. Now I'm just going to take a close look, make sure. Yeah, it's pretty self-explanatory. The only thing that I know students get a little tricked up is on the summary questions on page 303 and 304. You're basically writing a scientific summary based on the observation that was introduced in the workbook. So this rock pocket mouse population. So when you're writing your, for example, description of how the population has changed, you're applying it to this experiment with the rock pocket mouse populations. Okay, so use what you learned from this population study and apply it to your answering of page 303 and 304. Okay. So go ahead and you'll find your assigned questions that you're responding to, and then post those answers no later than Sunday midnight. Okay. The next assignment is we're going to kind of apply what we learned about natural selection to human population in response to malaria. So you'll start out by watching this video on malaria, which is a Disney video, and it's actually it's kind of funny because this is a public health announcement, and you can kind of think about if we had some vector like this rampant in our neighborhoods, would we do as destructive response as what was introduced in this video? So I just see the spraying of chemicals on people to kill mosquitoes, and I worry about the ecological impacts. So you can watch it, it's pretty funny. And then we have some information that we've given you. So we have the CDC information website, the history of malaria, and then how it's currently transmitted and so forth. So you'll use that information to help complete the pages 305 to 307. Okay? So that's all based on interpreting graphs, as well as using the resources we provided. So on page 305, you see these two graphs. So this gives you an opportunity to learn how to interpret them. Same with on page 307, we have another graph that you're going to kind of interpret. And then based on that information, you'll continue with learning about malaria and sickle cell anemia by doing this little activity. So in this activity, starting on page 307, you will need to perform a little experiment on your own using some materials. Now in the materials section on page 308, it says 75 red beans, 25 white beans. If you don't have red beans and white beans, use anything, okay? Basically, you could do erasers and paper clips. You could do, beads, different colored beads. Whatever works for you, okay? So you just need two sources or two types. You need a lot of one type and a little of the other. And then you're going to kind of use those to create your own little experiment with the allele frequency. Okay. And then once you've done that, you will go ahead and post your results in your workbook, and then answer the questions on page 311 and 312 based on your results. Okay, so it's basically this we're simulating the incidence of sickle cell allele within a population. Okay, so once you get that understanding, you'll post your answers to your assigned questions in the discussion board post. And then the final, oh, and by the way, that was due on next Tuesday, midnight. And then the final activity for the term, this is it. This is the last one. You will be looking at artificial selection and applying it to emerging diseases, so the evolution of antibiotic-resistant bacteria. So in this activity, you're going to need to create, you're going to need some materials again to kind of create this little scenario. So if you have beads, different colored beads, that would work perfectly in this experiment. If you don't have beads, if you have anything that has different colors to it, so, you know, if you have a bunch of different colored beans, like you have like the 21 bean mix or something like that, or you can even just cut out like pieces of paper of different colors and use that instead. Okay, but you will need to have, if you look on page 313, you're going to create this population of different bacteria representing different colors. Okay, so once you do that, then you're going to draw a graph. Okay, so on the very next page, on page 314, you're going to draw a graph of what that population is showing. Okay, so if you look at page 313, the original population had 2 lavender bacteria, so that's the least resistant. Okay, so on your x-axis, you're going to have the different color bacteria going from least resistant towards the y-axis, or the 0, to the most resistant further on your horizontal line. So you'll just write lavender, orange, pink, clear, white, etc. on the x-axis of your graph. Okay, and then on the y-axis, you're going to be, each of those beads represent 1,000 bacterial cells. So you're on the y-axis, you're going to be looking at the number of bacteria. Okay, and so once you have your graph set up with the x-axis being the types of bacteria going from least to most resistant, and then the y-axis with the population of bacteria, then you're going to graph it. Now, since these bacteria are different, are we going to do a line graph or a bar graph? Can we connect the dots between 2 species of bacteria? No. So this is going to be a bar graph because lavender bacteria are different from orange bacteria. You cannot connect the dots and get like a hybrid lavender-orange bacteria. Okay, so this will be a bar graph, and so you'll be creating your graph showing the population of each type of bacteria. Okay, so you use that as a starting point. Then you're going to be learning about how antibiotics work. And then in this particular scenario, you have 2 people, one of whom has done the antibiotics correctly, and the other who maybe did not follow the directions of her antibiotics correctly. And so she might have some issues regarding that. And so you're going to follow the directions, and you're going to, use your bacteria population of bacteria beads or colored pieces of paper or whatnot, and you're going to do this little activity to count how many of the bacteria ended up dying off from the antibiotics and how many survived, and then what happened to the surviving bacteria. Okay, so once you go through this scenario and collect your data, you're going to be putting the data in a table on page 316, and then you're going to create another graph, okay? So this other graph, which you have extra graph paper at the end of your workbook, by the way, so you can use one of those extra pieces of graph paper to plot the graph, or just use the original graph that you did on page 314 and add the extra results to it, okay? So you have the original population of Karen's sore throat population, and then you can add the population after the treatment, okay? So what happened to the population? And then you can do the same for Susie's. And you can see with all of this data on the graph, you know, what happened to the person who had read the directions and followed the directions about how to use antibiotics, and what happened to the person that did not follow directions carefully, and how that led to an increase in the most resistant bacteria population surviving, okay? So you'll answer those questions on page 319 and 320 based on your graph that you've created, and you're going to actually be posting that final graph showing Karen and Susie's sore throat populations. So again, that could just be all the data on written on one graph on page 311 or 314, or you can, you know, come up with a new graph if you fail. We have plenty of graph paper at the end of the workbook to work with. So you'll be posting that final graph as well as the answers to your assigned questions by Tuesday midnight. Okay. Now, it seems like we don't have a lot going on this week. And the reason being is that you will be completing your genetic diseases integration project and posting your presentation by Monday midnight. Okay, so next Monday. And so you'll take the rest of this week and finish off your preparation for this presentation that you're creating. So, you know, it just says here that you'll be posting your project no later than Monday, March 11th. So this is a hard deadline. I will not accept presentations that are posted next Tuesday or next Wednesday or next Thursday. And the reason being is that we have, we're posting these presentations for our fellow classmates to read and critique and ask questions about. Okay, so you do need to submit your project on time next week. That's why we have kind of an easier workload this week to give you plenty of time to finish up your project. And by the way, where do I get information about what to include in my project? I go back to topic 4. Down at the bottom is the integration project selection. That's where, you know, we have our assigned topics. This is where you will, you know, look closely at the integration project handout and make sure that you have addressed all of these parts in your presentation. Okay, so, and I believe I actually included, oh, maybe not. I will post a video on that as well. Okay, so this week you have 3 assignments plus a topic 7 quiz. And then you should be spending the remainder of the week working through your presentation that's due Monday midnight. Okay? If you have any questions, feel free to email me. Otherwise, good luck.